let's go ahead and get started. Welcome back. Okay, so this should be the last section of chapter 25. So I told you I was going to dig up a picture of an endospore, so this is what we have. So here's our bacteria, and within this bacteria, so endo means inside, we've got this resistant spore. So if the environment became very, very hostile, or if there just was no food or something and the colony died, these endospores would be able to be in a resting state for many, many years even, and then come back and regenerate the colony when conditions improve. So just wanted to show you an image of that. Okay, so let's talk about ecological diversity in prokaryotes, because it's tremendous. So first of all, they had quite a evolutionary head start. They've been on the planet for over 3.5 billion years, and they reproduce very quickly. And so they're able to evolve much more quickly. So not surprisingly, they have evolved to fill pretty much every ecological niche you can imagine on the Earth. And this becomes evident in a few different ways when we look at nutritional adaptations, when we look at some of their metabolic adaptations. And although they're unicellular, we see tremendous adaptations that allow cooperation among members of the same species, or sometimes even members of different species. So they're really, they really do it all. So we're going to talk first about nutritional adaptations. We really, every possible method of nutrition is present in prokaryotes, and not only that, but two of the four possible nutritional categories, the photoheterotroph, I'm going to use a different color here, the photoheterotroph and the chemoautotroph are only occupied by prokaryotes. So let's break down what these mean. So when we talk about nutritional adaptations, there's how you make your energy, so how you make your ATP, in other words, and what you use for a carbon source. What are you using to build your structure, your organic structures of your body? So for a photoautotroph, that would be something like a plant, or a cyanobacteria would also be a photoautotroph, excuse me. They get their energy from light, and all of this is in the table if you can't read my writing, and their carbon comes from CO2. So everything is inorganic. They're very independent. They don't need another organism's organic tissues, right? They can just use light from the sun and CO2 from the atmosphere. A photoheterotroph uses light as an energy source, but must also consume organic forms of carbon in order to build its structures. So, and photoheterotrophs are pretty rare. And we just find them in a couple of prokaryotes. There's also what's called a chemoautotroph. So they get their carbon source inorganically from CO2, and but they don't get their energy from the sun like a photoautotroph does. That chemo stands for chemical, so they're using some different inorganic substance, maybe sulfur or some other chemical, as their inorganic substance. So again, this is a very independent group of organisms. They don't rely on a different organism for any kind of organic tissue. And then there's the most dependent of all, the chemoheterotrophs, which is the category we fall into. We have to get our energy organically by eating other stuff, and we must get our carbon building blocks organically by eating other stuff. So those are the nutritional adaptations, and we see them all in the prokaryotes. They're pretty amazing. When we get into talking about animals, you'll see that it becomes much less diverse. Pretty much everybody's a chemoheterotroph. So let's talk about metabolic adaptations. Okay, so metabolic, nope, I don't want to be on cooperation, sorry. And you might be wondering, why are you showing me this picture of a plant's roots? Well, I'm showing you the picture of the plant root because of these little nodules on them that actually house bacteria that are capable of nitrogen fixation. So under this category of metabolic adaptations, I want to talk about both oxygen use and nitrogen fixation. Okay, so we're going to talk about those two categories. So starting with oxygen use, there's a couple of different adaptations we see in prokaryotes regarding oxygen use. We have what are called obligate aerobes, which must use oxygen for cellular respiration. Okay, oxygen, not optional. They must have it. There are also what we call obligate anaerobes. So, no O2 needed. And in fact, O2 is toxic to them. So, there's a couple different ways to do your cellular respiration without oxygen. You might be doing fermentation. So, for example, you might be, if you think about the microorganisms that are able to use perhaps alcohol or lactic acid, that would be an example of fermentation. Or they might do anaerobic respiration, where they use something other than oxygen as the final electron acceptor in the electron transport chain. And if you have no idea what I'm talking about and you need a refresher on cell respiration, you can talk to me about that in lab if you like. Hopefully you know what I'm talking about when I say electron transport chain and fermentation. So there's also what we call facultative anaerobes. So we have facultative and they can kind of use oxygen or not. They can use oxygen when present, but use some other kind of, use one of the anaerobic pathways, such as fermentation, when O2 is absent. So they have some flexibility there. So we also have metabolic adaptations regarding nitrogen use. Eukaryotes can use very few forms of nitrogen. So we can take in basically nitrogen that has been, converted and we get from the tissues of other organisms. But some prokaryotes can do nitrogen fixation, which I mentioned is when you convert atmospheric nitrogen, or N2, into an organic form such as ammonia or something like that. And then these organic forms of nitrogen can be used by other organisms. And some species enter a symbiotic relationship with plants, where the bacteria provides nitrogen fixation, fixation, and the plant provides shelter and carbohydrate from photosynthesis. So this would be a great example of a mutualism, right? So in these nodules on this root exist bacteria that are capable of nitrogen fixation. They can take inorganic nitrogen and transform it into an organic form that the plant can use. In turn, the plant gives it a place to live and provides it with sugars from the process of photosynthesis. So it's this lovely mutualistic symbiotic relationship between the two different species. We also can get some adaptations involving cooperation. So I talked about this a little bit already when I talked about the colony that we get in some cyanobacteria, such as that Anabaena. So some forms of cooperation, for example, the vegetative cells make photosynthesis products, the heterocyst makes nitrogen products, and then they can share and cooperate. So some species have these specialized cells, like the heterocyst or the vegetative cells, that basically perform only one function, and then they can sort of share their products around. So other species create what are called, so we have these sort of, the heteromorphic cells in the colony that cooperate by sharing products. So, for example, the Anabaena. Another form of metabolic cooperation we see are in biofilms. So a biofilm is a surface coating colonies of several species of bacteria. A surface coating film made of several species of bacteria. So it's kind of a cool thing. It's sort of a living kind of community where we have several bacterial species aggregating together, usually secreting some kind of sticky meshwork that holds them together. And probably the most well-known biofilm is dental plaque. So that is what dental plaque is. Basically a community of bacteria that have created this thin film. And I don't know if you remember those little tabs sometimes a dentist would give you that would stain your plaque purple, but that's what you're looking at. And then here is an SEM of dental plaque. So you can actually see how delicious it really looks in real life. Okay, so finally I want to talk a little bit about some of the ecological roles of bacteria. So some bacteria, actually most bacteria are actually good. We tend to give them a bad rap and think of them as, the enemy, and indeed some can cause disease, but most of them are good for us. So symbiotic bacteria means bacteria that's living in close proximity to us, so in or on us. So hundreds of species, by the way, sometimes I abbreviate species by SSP, live in or on us. And in terms of sheer numbers, there are many more individual bacterial cells in and on our bodies than there are human cells. And I love this figure because it kind of shows 70 to 90%, or about 100 trillion of the cells that make us up are actually bacterial cells, with only about 30 trillion being human cells. So we're pretty much just bags carrying around bacteria. Most are unstudied species and we don't know their function. They may have a function, but we just don't know it yet. So many bacteria are actually mutualistic, meaning they help us somehow. So we have about 500 to 1,000 or so species in our gut that help us acquire nutrients, that help us, for example, unlock vitamins from nutrients we otherwise wouldn't be able to get, help us digest our food, break things down. They're very, very important to the functioning of our gut. And we probably have many, many, many more mutualistic species than this. We just don't know about all of them yet. But some bacteria are what we would call pathogenic bacteria. A pathogenic bacteria is one that causes disease. And there are two kind of main ways they do this. They produce diseases in us by releasing toxins, and there's two sort of types of toxins, what we call exotoxins and what we call endotoxins. So an exotoxin is a soluble protein. So it's a protein of the bacteria that is released by living, multiplying bacteria. Okay, so these guys are living and doing their thing, and these exotoxins are being secreted from them. They are proteins coming from a living bacteria. So some examples of exotoxins, let me just get this out of the way for a minute, would be produced by cholera. Cholera produces exotoxins. The bacteria Clostridium botulinum, which causes botulism, though that's caused by an exotoxin. Anthrax, so Bacillus anthracis, the disease, or I'm sorry, the bacteria causing anthrax, that is caused by an exotoxin secreted by the living bacteria. But then there are also those that there are also endotoxins. So endotoxins are liposaccharides that are released by bacteria when the cell either grows or when it dies. So when this bacterial cell dies and the cell wall breaks up, these liposaccharides can be released. And in some cases, when the bacteria grows and if the wall breaks up, it can be released. Now, I don't know if you remember, but I said that the gram-negative bacteria tend to have these liposaccharides. So not surprisingly, we're going to get a lot of gram-negative bacteria doing this. So examples would be E. coli or Salmonella are two types of bacteria that have the endotoxins that can be released and are what will make you sick. Okay, so we're finally at the end of our chapter 25. I know it was long, but thanks for hanging in there with me. If you have any questions, please feel free to email me anytime, and I'll see you in lab.